For more than a century, the story of human origins has been told as a story of expansion and success. A small population of early modern humans emerged in Africa, developed new tools and behaviors, and gradually spread across the globe. Along the way, they adapted to deserts, forests, ice ages, and coastlines. They learned to hunt efficiently, to communicate symbolically, and to survive in almost every environment on Earth. This narrative has long suggested resilience flexib. Yet within this success story, there has always been a quiet puzzle. When scientists compare humans today from distant continents and vastly different cultures, they find something unexpected, far more similar than most other large mammals. Far more similar than our ancient relatives once were, for a species that now numbers in the billions and occupies every corner of the planet, our gen for decades. This fact was acknowledged but poorly understood. It was explained loosely through ideas of recent origin, population expansion, or cultural dominance. Archaeology provided tools, bones, and settlement patterns. Fossils revealed changing anatomy. Artifacts showed innovation and migration. But none of these could fully explain why so much genetic variation appears to be missing. The turning point did not come from a new fossil discovery or a revised timeline. It came from something far more fragile and far more powerful. Ancient DNA, preserved in bone, tooth, and sediment, began to offer a direct biological record of past populations. For the first time, scientists were no longer limited to interpreting shapes and tools. They could compare genetic sequences across tens of thousands of years, across vanished populations. What emerged from this genetic record was not a simple story of steady growth. It was a story shaped by loss, isolation, survival, through narrow corridors of time, and repeated moments when humanity came close to disappearing altogether. A story in which entire populations vanished. A story that helps explain why nearly everyone alive today shares such a small slice of humanity's ancient diversity. To understand why our genetic inheritance is so limited, we must first understand what we thought we knew before DNA began to speak for the past. Before ancient DNA entered the picture, the story of human genetic diversity was built almost entirely from visible remains and living populations. Anthropologists compared skull shapes, body proportions, and skeletal features across regions. Archaeologists traced stone tools, artifice ornaments, and burial practices. Geneticists, working only with modern DNA, compared blood types and later small sections of the genome. Together, these approaches formed a coherent and confident framework. In this traditional view, modern humans emerged in Africa. Over time, population size slowly increased. Small groups left Africa, in waves, spreading into Eurasia. As these groups expanded, they diversified culturally and physically, but they remained part of a continuous human lineage. Genetic differences between regions were assumed to reflect adaptation to local environments over long periods of time. Limited genetic diversity was interpreted mainly as a consequence of recency. Humans, it was thought, simply had not existed long enough to accumulate deep genetic differences. Compared to species that had been evolving for millions of years, Homo sapiens were newcomers. This explanation was neat, logical, and largely uncontested. If it comes alongside from this framework, human populations were believed to have expanded steadily with occasional local setbacks, but no major global collapses. Extinction, when it occurred, was something that happened to other species. Neanderthals vanished, Denisovans disappeared. Earlier Homo species faded into the fossil record. Modern humans, by contrast, were seen as persistent and resilient. When population bottlenecks were discussed, they were usually framed as regional or brief. A drought might reduce numbers in one area. A volcanic eruption could disrupt a migration route. But these were considered interruption, not defining features of human history. The overall trajectory was upward. Mitochondrial DNA pointed to a common maternal ancestor in Africa. E-chromosome studies suggested a similar pattern for paternal lineages. These findings supported the idea of a recent African origin followed by rapid expansion. The low genetic diversity observed in living humans was seen as a natural outcome of this process. Yet there were warning signs that this explanation was incomplete. When geneticists compared humans to other primates, the contrast was striking. Chimpanzees living in a single forest region often showed more genetic variation than all humans worldwide. This was difficult to reconcile with a simple story of recent origin alone. Something more severe than youth seemed to be missing from the explanation. There were also hints in the fossil record that did not fit neatly. Long gaps appeared in certain regions, Archaeological traditions vanished abruptly. Some areas show repeated abandonment and reoccupation. These patterns suggest effort at a global scale. Most importantly, all reconstructions relied on indirect inference. Fossils showed who might have lived where tools suggested how they behaved. Modern DNA offered only a snapshot of survivors. Entire populations that left no descendants were effectively genes to living people. Traditional methods had no way to detect them. This limitation shaped how the past was imagined. Human history appeared smoother than it truly was, filtered through the biology of those who survived. The assumption remained that our genetic uniformity was simply the result of a young, expanding species. That assumption would not survive the arrival of ancient DNA. The breakthrough began quietly with fragments so small they were once thought impossible to recover. Ancient DNA does not survive easily. Heat, moisture, bacteria, and time all work to destroy it. For decades, most researchers believed that DNA older than a few thousand years would be too damp were plagued by contamination and false results. The genetic past remained locked away. That changed in the late 20th and early 21st centuries. As laboratory techniques improved and standards became stricter, scientists learned to work in ultra-clean environments, to identify and filter the chemical damage that marks truly ancient genetic material. Advances in sequencing technology allowed researchers to read millions of tiny DNA fragments and reassemble them like pieces of a shattered manuscript. Crucially, researchers discovered that certain parts of the human body preserve DNA better than others. Dense bones, especially the inner ear region of the skull, known as the petrous bone, teeth, with their protected pulp cavities, also yielded genetic material. In some cases, even sediments from caves retain traces of DNA left behind by people who had lived there tens of thousands of years earlier. By the early 2010s, ancient yeomans were no longer rare curiosities. Scientists began suing entire golems from individuals who lived 40, 50, even 60,000 years ago. These were not reconstructions based on assumptions. They were direct biological records of real people who had once walked the landscape. The first major shock came from comparisons between ancient and modern humans. When early Eurasian yeomans were analyzed, they did not fit neatly into the genetic variation seen today. Some lineages present in ancient individuals had no close equivalents in living populations. Others showed levels in genetic diversity that seemed far higher than anything observed among modern humans. Even more striking were the results from Africa. For a long time, ancient DNA from tropical regions was considered unattainable because of poor preservation. But as techniques improved, genetic data began to emerge from ancient African individuals as well. These golems revealed deep population structure within Africa, with groups separated from one another for long periods of time. Some of these ancient populations were genetically distinct enough that, had they survived, modern human diversity would look very different. At the same time, ancient DNA illuminated interactions with other human groups. Neanderthal germs, sequenced from individuals who lived in Europe and Western Asia. Denisovans, known initially from only a few fragments found in Siberia, turned out to represent multiple deeply divergent lineages. When modern human germs were compared to these ancient ones, clear signs of interbreeding emerged, but interbreeding alone did not explain the central puzzle. While ancient humans once displayed considerable genetic variation, much of it vanished. Many ancient lineages left no trace in living people. Others survived only in small fragments. The genetic record showed repeated contractions in population size, followed by expansions from very limited pools of survivors. By examining patterns of shared mutations, scientists can estimate not the thousands or hundreds of thousands, but in some cases to numbers that suggest only a few thousand breeding individuals worldwide. These were not brief local events. The genetic signatures pointed to prolonged bottlenecks lasting thousands of years. During these periods, vast stretches of human genetic diversity were lost, not through selection or adaptation, but simply because the people who carried those genes did not survive. Ancient DNA transformed human history from a smooth expansion into a fragile sequence of survival episodes. It showed that the humanity we know today descends from a narrow genetic thread that passed through repeated filters of climate change, environmental stress, and demographic collapse. With this genetic evidence in hand, researchers could finally begin to explain why modern humans are so genetically similar. The answer was not just where we came from, but how close we came to disappearing along the way as ancient humans accumulated. A clearer and more unsettling picture began to emerge. Human genetic diversity was not always limited. In fact, for much of our early history, it appears to have been far greater than it is today. The reason modern humans share such a narrow genetic range is not because diversity never existed, but because so much of it was lost. One of the most important findings came from the reconstruction of ancient population sizes over time. By examining how often certain genetic variants appear and how they are distributed across groupings, scientists can estimate what is known as effective population size. This is not a head count of individuals, but a measure of how many people were contributing genes to future generations. When ancient DNA was used to extend these estimates deep into the past, the results showed repeated and severe reductions. These bottlenecks were not evenly spaced or mild. Several appear to have occurred during periods of major environments and development. Around 70,000 years ago, during a time of intense climatic fluctuation, for thousands of years the number of breeding individuals may have remained extremely low during this prolonged bottlenecks. Many genetic lineages simply disappeared. Another critical insight involved the structure of early human population. Ancient DNA revealed that early Homo sapiens were not a single unified group spreading outward. Some lived in ecological refuges, separated by deserts, forests, or changing coastlines. Others followed seasonal routes that rarely overlapped with neighboring groups. Yet exchange between these populations was limited when environment is. Small, isolated populations are especially vulnerable to extinction. If a drought, cold period, or resource collapse struck one region, the people living there might vanish entirely. Ancient DNA shows that this happened repeatedly. Whole populations disappeared without leaving descendants. The genetic record also revealed that later expansions were often built on extremely narrow. When conditions improved and humans spread into new territories, they frequently did so from small survivor groups. These expansions amplified a limited subset of genetic variation while permanently excluding what had only a fraction of humanity's former diversity. Interbreeding with other human groups added complexity, but did not reverse the overall pattern. When modern humans encountered Neanderthals and Denisovans, they absorbed some of their genes. However, these contributions were small compared to the diversity that had already been erased within Homo sapiens itself. Ancient DNA shows that Neanderthals and Denisovans were also genetically diverse in the past, and they too experienced population declines. In some regions, ancient neons reveal modern humans replacing earlier human populations with little or no genetic continuity. In parts of Eurasia, early Homo sapiens groups appear briefly in the record, only to vanish and be replaced by later migrations from different source populations. Perhaps the most striking revelation is how recent much of today's genetic uniformity is. Many of the lineages that dominate modern human germ appear to trace back to expansions that occurred within the last 40 to 50,000 years. This means that the genetic similarity shared by people across the world today is not a deep ancient feature of our species, but the result of relatively recent survival and spread. Ancient DNA also helped explain why modern humans differ so little from one another, despite cultural and geographic separation. The populations that gave rise to today's world were already genetically constrained before they expanded widely. As a result, even when humans adapted to new environments, they did so using a limited genetics toolkit inherited from a small group of ancestors. Taken together, these findings overturned the earlier assumption of steady growth and continuity. Human history, as written in DNA, is marked by repeated narrowing events. Diversity was not slowly accumulated and preserved. It was repeatedly stripped away, leaving behind a lineage shaped as much by loss as by success. The realization that humanity passed through repeated genetic bottlenecks changes how we understand ourselves, not as an abstract species, but as a shared biological lineage. Ancient DNA reveals that the people alive today are not representatives of all that humanity once was. We are the descendants of those who endured a series of narrow survival passages, each one trimming away entire branches of our past. This does not mean that ancient humans were fragile or unsuccessful. On the contrary, the fact that any lineage survived such pressures speaks to adaptability and resilience. Many populations, likely well adapted to their local environments, did not persist. Their disappearance was not necessarily due to inferiority or failure. Often it was simply the result of isolation combined with sudden invective. This history explains why humans today share such a strong biological similarity across continents. Our differences in appearance and culture emerged on top of a foundation that was already genetically narrow: skin, pigmentation, facial features, and physiologemation, responsigenetic separation. Beneath these differences lies a common inheritance shaped by a small number of ancestors. Ancient DNA also reframes how we think about human unity. Genetic similarity is not evidence of uniformity in experience or culture, but it is evidence of shared descent through hardship. The gemens of people living today carry the imprint of ancient population collapses that affected everyone's ancestors, regardless of where they live now. In this sense, modern humanity is biologically interconnected not just by origin, but by survival. These findings also clarify why certain genetic diseases and maladies are widespread. When populations pass through bottlenecks, rare genetic variants can become common simply by chance. Traits that might have remained uncommon in a large diverse population can spread rapidly when the population is small. Ancient DNA helps trace the timing and context of these shifts, grounding modern health patterns in deep history. Importantly, this research does not reduce humanity to genetics alone. Culture, language, and social organization have always played critical roles in survival, but ancient DNA shows that biology set the boundaries within which culture operated. When populations shrank, cultural knowledge may have been lost along with genetic lineages. When populations expanded from small groups, cultural traditions spread alongside a limited genetic base. Perhaps the most profound consequence of these discoveries. Genetic evidence makes it clear that no living population represents a more ancient or more complete version of humanity than any other. Everyone alive today descends from the same constrained genetic pool, shaped by the same bottlenecks and expansions. In this light, limited genetic diversity is not a flaw or a weakness. It is a record of endurance. It reflects a history in which survival depended on cooperation, mobility, and adaptation under extreme pressure. Ancient DNA does not diminish humanity's past. It reveals how close that past came to ending, and how much of it had to be lost for the present to exist. For most of human history, our understanding of the past was shaped by what endured: bones that fossilized, tools that survived burial, and cultures that left visible traces. Ancient DNA has shifted that perspective. It has shown that absence is just as important as presence. What is missing from our gemens, the limited genetic diversity shared by humans today, is not a simple consequence of youth or success. It is the result of a long sequence of constraints, collapses, and recoveries. Again and again, human populations were reduced to small numbers. Again and again, survival depended on a fraction of the people who had once existed. Each recovery carried humanity forward, but it also carried forward less of what had come before. Ancient DNA provides a direct connection to those lost populations. It allows us to see that the past was not a straight line leading inevitably to the present. It was a branching, fragile process in which most branches did not endure. The people alive today represent a narrow passage through that history, not its full breadth. This understanding does not diminish humanity's achievements. It places them in context. Cultural complexity, global expansion, and technological innovation arose from populations that had already survived. The genetic similarities that unite humans today are a biological reflection of shared endurance, rather than sameness of experience. As ancient DNA research continues, the picture will become more detailed, but its central message is unlikely to change. Human history is not defined solely by growth and expansion. It is defined equally by survival through loss. Our gemens are records of that survival, carrying the imprint of ancient bottlenecks that shaped who could pass forward into the future. In listening carefully to this genetic record, we gain a clearer view of humanity's past, one grounded not in myth or assumption, but in the quiet persistence of DNA itself.